All right, let's go ahead and get started. Sorry for making you wait a couple minutes. I was trying to fix a couple technical difficulties on my end. I meant to upload last class's video lecture. I did not have the aspect ratio set up properly between my MacBook and the system here, so the video is a little finicky. I'm trying to finish editing it. I don't think it'll work, so it might just be a really weird looking video that you'll hear my voice, but you can also go through your own slides as you're listening to it. But I think I fixed it today, so I'll make sure that works. Before we get started, we have two of our other TAs for the course that I want to try and point out so you guys know their face and know them when you're going to office hours, or who to reach out to if you need help with something beyond me or the others. We have Taylor, who's a graduate TA right here, PhD student in Jeff Sauceman's lab, brilliant guy and will be willing to help with anything. We have Catherine Taylor over there in the back. She TA'd with me last year. She's great, and I think you guys are privileged to have a really qualified and solid teaching team to help fill in the gaps that I might mess up along the way. But with that being said, we can go ahead and get started. So for today's lecture, we're going to begin looking at amino acid structures and their function and how they relate to protein function and structure. We're going to try and consider the evolutionary constraints that tie into amino acids and the disease that we're going to be focusing on today, which is sickle cell disease. And we're going to be looking at the history, background of sickle cell disease, try and look at the learn more about amino acids, how they relate to protein structure and function, and then tie back to sickle cell disease and look at the clinical application of this all. And so we'll begin by looking at the etiology of sickle cell disease. So as I mentioned in the last lecture, words that are in blue are words that you're going to try and take note of and understand what they mean. So etiology means the cause. So we're first going to be looking at what the cause of sickle cell disease is. So sickle cell disease is a recessive inherited blood disorder. Recessive, what that means here is we get two copies of genes from each of our parents, and a recessive disease means that you need to have both copies inherited. Usually dominant disease typically means that you just need one copy. So recessive means, if you remember back to your Punnett square that you did probably years ago, for recessive, you need to have both alleles. And this is caused by a mutation in one gene, which means it's a monogenic disease. Diseases that require multiple genes to be mutated is a polygenic disease. So this is a recessive monogenic disease that affects the hemoglobin beta chain gene. For this course, as well as moving forward, it's helpful to understand the naming classification for genes. And so the goal is not, the reason why HBB is the gene name for the beta chain is highlighted in blue, not because I want you to remember every single gene that we cover or to specifically remember that one, but rather appreciate the naming nomenclature for genes, that it's usually, for human genes, all capitalized, italicized. So the symptoms for sickle cell disease. So what ends up happening is the hemoglobin undergoes a conformational change where it looks like almost like the sickle that you might have seen pictures of a Grim Reaper having. And what ends up happening is that you'll get the red blood cells beginning to, they're the ones that undergo this change, and they'll begin to stack up and cluster together, occluding capillaries, and they'll also lyse, releasing their contents. And when they're releasing their contents, they're releasing a ton of iron, if you remember from Physiology 1. And bacteria love iron, and so this leads to infections. So sickle erythrocytes are another name for red blood cells. You'll also have anemia, which is a loss of red blood cells because these red blood cells are lysing because of the change in the hemoglobin. You'll have acute chest syndrome, so the capillaries in our lungs are going to become occluded because of these erythrocytes stacking up together and blocking up these small blood vessels. So typically, red blood cells are really pliable and are able to squeeze through these capillaries that are in our lungs, that are in our digits. And so when they lose this pliability, they begin to just stack up together and occlude. Like I mentioned earlier, when they lyse or burst, they're releasing iron, and bacteria love iron. And so you'll get frequent infections. And you'll also get dactylitis, which is swelling of the fingers or digits. Typically, whenever you see something with itis on the end, it typically means inflammation of. And so in this case, dactyl is usually referring to digits. And so to understand a little bit more about the epidemiology of sickle cell disease, we're going to look at the prevalence and the incidence. Prevalence typically refers to how many people have it now. It's an assessment of the current moment of how many people in the country or in the world have this disease. And so as of now, there are 90 to 100,000 cases in the US. So that's the prevalence of sickle cell disease. Incidence is looking at the probability or the statistics of it happening in the future. So it's looking ahead of what the likelihood is of this disease occurring within a population. And so the sickle cell disease primarily affects African Americans from West African descent, and it happens in 1 in 500 births. And we'll actually take a moment towards the end of the lecture to actually explain why it emerges in West African populations. So now pivoting to the looking at peptides and looking at, and then we'll turn back to how this relates to sickle cell. We're going to start with a couple of slides that were in your pre-lecture just to try and get on the same page. So amino acids are the building blocks of proteins. When you string together multiple amino acids, you're making a peptide. And so the way amino acids and the way peptides are read are from the N terminus to the C terminus. The N terminus, also the amine group to the left here, and you read it over to the C terminus or the carboxyl group. So you always want to make sure that when you're reading it, and we'll have an exercise later today to try and illustrate this, you always want to make sure that you're looking at, that you see the amine group that then goes to the alpha carbon with that side chain alpha carbon R1, and then that goes to the carboxyl group. And so you'll string these together, and we'll talk a little bit later about how these amino acids are strung together, how the bond that holds them together. But whenever you're reading a amino acid, you're always reading, or a protein, you're always reading from the N terminus, the amine group, to the C terminus, to the carboxyl group. And the way that these amino acids are strung together or held together are through what's called a peptide bond. And so these peptide bonds, the bond occurs between an amine group of one amino acid, or sorry, forgive me, the carboxyl group of one amino acid to the amine of another. And so if you'll see here, we have our amine group, the alpha carbon with the side chain to the carboxyl group. And then here's the peptide bond that binds to the next amino acid's amine group. And so what you'll be able to see is that you'll have a polypeptide where you'll have one amino acid here, another amino acid here. And so what's important to know is that this peptide bond is rigid or planar. And so the bonds that we have, yes? So the peptide bond is what is a kind of amide bond. So this peptide bond is rigid and planar due to the resonance. And so it's oftentimes these bonds dictate the structure or folding of different of how amino acids relate together. And so here, what's important to know is that this peptide bond is rigid and planar. So we are next going to talk about different hierarchical organizations of amino acids. So we're going to start with primary structures, and then we'll jump to tertiary and quaternary, and then go back to secondary. So what's important to know here is that primary structure is just the list of the amino acids. So we have methionine, valine, histidine here. So it's not, you don't call it 1 degree structure. That's just a shorthand for primary. So the primary structure, again, is just a listing out of the amino acids that make up a specific protein. And what's important to also consider is thinking about the conservation or the preservation of a specific amino acid sequence across time, and a way that we can do that is look at different species. So here we have, from top to bottom, we have humans, rhesus macaque, the cow, sheep, boar, I think, bat, mouse, chicken, and crocodile. And so this is the beta chain, the gene for beta chain of hemoglobin. And so what's really helpful to consider is that from humans down to chicken, we have about 150 million years of evolution. And so what's really interesting to consider is that we have sequences that are conserved across those millions of years. The sites of high conservation are in blue and purple. And so it's helpful to think about, when looking at proteins of their primary structure, how things have changed over time and how things have been conserved. And if something's been conserved over time, that highlights its importance, that it's been selected for over millions of years. And so we have the conservation of proteins over time across species, but we also have the customization of protein sequences. And so what we have are different isoforms of proteins. Those are usually different, you can call them different, almost like species of a protein. It's kind of a different type of the protein. And so with hemoglobin, we have the beta chain from top to bottom of HBB, D, E, and A. We have the beta chain, the delta chain, the epsilon chain, and the alpha chain. And so there are 4 different types of hemoglobin proteins. And there's pretty high conservation of them across, but there's also customization of them over time. And so what's believed to have happened is that there was one ancestral protein of hemoglobin, and over time it was duplicated to have 2 types of, to have 2 hemoglobin genes. And that was selected for over time because it was evolutionary advantageous. And then that 2 became 4 after another doubling. And so over time, what happened is that it became more advantageous for organisms to have 4 types, and that's how we have what we have today. And if you have any questions, please raise your hands and interrupt. Happy to answer any questions. So we have our amino acid table that was also in your pre-lecture. What's important to know is that I do not want you to exert your mental bandwidth. I don't want you to dedicate it towards memorizing all of this, but rather what we want for you is to develop an associative understanding of the different groups of amino acids and how moving forward that can inform the different bonds that we're going to discuss, as well as how it can inform specific proteins and their structure and their folding. And so the important part of protein folding is that we discuss the primary structure being the list of the amino acids that make up a protein, but proteins do not exist as a spaghetti string. We look at and consider them in their primary structure, but proteins actually exist in a folded state that's informed by the different bonds that exist as a result of the amino acids that we see. And what we're going to try and do, in today's lecture is try and understand this and apply that back to sickle cell disease and hemoglobin. So one of the first things I want you to consider is that there are one of the first groups of amino acids we want to try and discuss and look at today are the sulfur-containing amino acids, primarily the first one being cysteine. So this has a sulfur group on the side chain here, and as well as methionine. But what's important to know is that these cysteines are really important in creating what's called disulfide bonds. So a disulfide bond is when 2 sulfurs are bound together as a covalent bond. And what's important to know is that this bond occurs or changes depending on the environment that it's in. So in a reducing environment, what you'll see is that the 2 cysteines are, it's more thermodynamically, the thermodynamic equilibrium is such that in a reducing environment, they're not going to want to try and bind there. But in an oxidative environment, and we're going to try and discuss a little bit later today, and what do those environments look like in a cell or in clinical setting. In an oxidating environment, you're going to lose the 2 electrons, lose the 2 hydrogens, and see that a disulfide bond is created. And so one of the reducing environments that we see is in the cytoplasm. And that's, again, it's advantageous for that to be the case, because with proteins that exist in the cytoplasm, you don't want to see these disulfide bonds occurring, but rather those that need disulfide bonds the cysteines to create a disulfide bond. The oxidizing environments that we see are outside of the cell, outside of the cytoplasm, in the extracellular space, or in the interior of many organelles. And some of those organelles are, for example, the Golgi apparatus or the endoplasmic reticulum, the ER. And that makes sense because those are the sites of protein synthesis and protein modification. And so the proteins that need to have disulfide bonds to occur, those are the sites where it would happen. So as an example, hemoglobin does not have any disulfide bonds. So instead, we're going to look at insulin. And so if you think about insulin and where it is present in the body, it's in the extracellular space. It's in the bloodstream. And so in that oxidative environment, you have 2 cysteines. You have 2 sites. You have 3 sites. Forgive me. 3 sites of disulfide bonds. You have disulfide bonds here across the 2 chains. You have it on the same chain down here. And then towards the end, you also have, again, across the chains, a disulfide bond there as well. And that's important because it permits insulin to fold in such a way to be in its bioactive form. So without these disulfide bonds, insulin would not be bioactive. And so this is, again, trying to illustrate that the bonds that we see present as a result of those amino acids here help inform the structure and the function of the protein. Another bond that is important in considering how amino acids inform protein structure and function is electrostatic or ionic bonds. This involves a transferring of electrons between the atoms rather than sharing, which is covalent bonds. And so what's important to know is that this is a result, that this requires charges to be shared. But then when you have salts, for example, if you added sodium chloride to a location, to a milieu, those salts would disrupt the transferring of electrons, and thus disrupt the protein structure. And so sometimes when doing experiments, if you want to try and break down a protein, you'll actually put it in a salt-rich environment that then allows you to try and ask certain questions or test certain things. So for our amino acid groups that have a charge, these are positively charged amino acids. We have arginine and lysine. Histidine with a pKa of 6 can sometimes be positively charged. It just depends on the pH. But the two that are important to remember here that are positively charged are arginine and lysine. And our acidic side groups that are negatively charged are aspartate or glutamate. You might also hear them referred to as aspartic acid or glutamic acid. So now we can look at these amino acids in the context of hemoglobin. On the left here, we have a surface map of a hemoglobin macromolecular protein. And I say macromolecular protein because hemoglobin is a tetramer, which means it has 4 protein subunits that come together to form hemoglobin. And it's also a heterotetramer, which means that it's a mixture of different kinds of hemoglobin subunits that come together. So what that looks like here is that you have 2 alphas and 2 betas that make up a hemoglobin protein here. Bless you. And what's important to know here is that the tertiary structure of a hemoglobin protein, so forgive me, I'll take a step back. So hemoglobin is a combination of hemoglobin proteins that come together, which means that if you had a jar of pure beta chain hemoglobin, it would be able to undergo folding and exist as its own protein. And so the tertiary structure here for proteins are either the specific units, a protein that can exist on its own. The quaternary structure is when all of the macromolecular form of that protein comes together. So we can look at the beta chain tertiary structure or the entire hemoglobin quaternary structure when all these subunits come together. Any questions? All right. And what you can appreciate also is that there's the protoporphyrin ring, those these red structures embedded within the hemoglobin. That's what when in physiology 1, when talking about the function of hemoglobin, how it contributes to oxygen transport, that's what those are in there. They're embedded within the hemoglobin there. And again, the point I want to try and make sure that comes across clearly is that each subunit of hemoglobin, alpha, beta, delta, or epsilon, is its own protein that undergoes folding and that can exist on its own. But hemoglobin, the macromolecular protein, is when all 4 subunits come together, 2 alphas or 2 betas. And to the right, yes. Yeah, so when we were looking back at the primary structure, it's the amino acid sequence that determines if it's an alpha, a beta, a delta, or epsilon. And so they each have their own gene that has a specific that elicits specific amino acids to be created, and then it undergoes folding when it's created to make either the beta unit or the alpha unit. And then what we're about to discuss in a moment is how those subunits come together to create the hemoglobin protein. And so to the right, what's happened here is in a computer model, the hemoglobin macromolecular protein has been split in half and pivoted so that you can see the insides of it. And the sites of red are the positively charged amino acids, and the sites in blue are the negatively charged amino acids. And what I want you to appreciate is that if you look in the highlighted squares, you can actually see that the sites that would again come together have positively charged on one side and negatively charged on the other side. And at another side, again, you have the positively charged and negatively charged, meaning that these sites will come together and have ionic bonds that help these subunits come together. And so it's held together. One of the ways in which this hemoglobin is held together is by ionic bonds because of the amino acids that are present within the different subunits that then come together, that then hold this entire molecule together. So to help illustrate a little bit more about how electrostatics affect protein structure and function, I want you to imagine this little yellow tennis ball is a protein, and it's going to enter into this spectrum, this jar that has a spectrum of pHs. And so in an acidic environment, what's going to end up happening is that it's a hydrogen-rich environment, and the negatively charged amino acids will then take on these protons, take on the hydrogens, and that's going to change the charge of the amino acid. It's going to change the charge and then eventually change the structure and function of this protein. And so it's going to neutralize the negative charge here on the amino acids. And so in an acidic environment, a protein is going to become positively charged. Now, if it slides down into an alkaline environment, that's a proton poor environment, okay? And so this alkaline environment will then, it's then going to become negatively charged because it's proton poor. They're going to be donating hydrogens to that site, and it's going to become negatively charged. What's important is that there's going to be a sweet spot, a little Goldilocks zone, where it's going to be an electrostatic equilibrium, where it won't be donating or taking on protons, but rather at equilibrium. And that point is called the isoelectric point. And so the pH at which that happens, forgive me, the pH at which a protein is at electrostatic equilibrium is called the isoelectric point. And that's really important to know because that helps inform us about that proteins, what is the balance of their positively charged and negatively charged amino acids, as well as when trying to handle that protein, what environment is it in equilibrium. And so, for example, the PI, the isoelectric point of hemoglobin, is 6.7. And so that means that it has slightly more positive, it has slightly more negatively charged amino acids. Are there any questions there? Yes. Yes, absolutely. It is different for different proteins. I think I saw a second hand. Yeah. Yes. So just to make sure I say this clearly, the PI, or the isoelectric point, is the pH at which that protein is at electrostatic equilibrium or electrostatically neutral. Any other questions? These are great questions, by the way. All right. So we'll take a step back, and now that we've done primary structure, tertiary structure, and quaternary structure, we'll now look at secondary structure. And secondary structure is informed, the local folding that happens as a result of secondary structure, or happens that we look at when we look at secondary structure, is a result of hydrogen bonding. And so if some of you might remember discussing dipoles in your chemistry classes, what the way hydrogen bonding occurs is that it's mediated by it's the attraction between an electronegative atom in one molecule and the hydrogen in another molecule. And so, for example, the classic example when considering or discussing dipoles is water. And so you have the hydrogen that has a negative, oxygen that has a negative dipole, and it's going to undergo hydrogen bonding to an adjacent water molecule, but to hydrogen from another molecule. And so over time, what will end up happening is, for amino acids, we actually see hydrogen bonding occurring between a carbonyl group and the hydrogen in an amine group of another amino acid. And so these are typically weaker forces, but this hydrogen bonding is really important when considering how a polypeptide sequence begins to take shape and begins to take form. There's two important structures, or I'll say structures. There's two important structures that hydrogen bonding gives rise to with polypeptide sequence. The first is the alpha helix. It's a result of hydrogen bonding that occurs vertically, and it's denoted by the dotted line. So you can see towards the top here, there's an oxygen from a carbonyl group towards the top that then has hydrogen bonding with the amine group below, the hydrogen to the amine group below. And so that contributes to how the alpha helix emerges. And usually for each turn, for each right-handed turn in an alpha helix, you have 3.6 residues per turn, residues meaning amino acids. And it's easiest to see, it's easiest to take note of the residues by looking at the side chains. And if you look at this side chains, you'll notice that all of them are pointing outwards. The side chains being denoted by the R. And so what this helps us to learn about a protein is that if all of them are pointed outwards, and if all of them are hydrophobic or hydrophilic, it helps inform not only what its interactions are with its microenvironment, but also potentially where in the cell, where in the cell it might go, or what it might do when it leaves the cell. So taking a look at hemoglobin, what you can see is, what I should say is the ribbons that you see on the right image. So what we've done is, this was the same surface map from the previous slide. We've just kind of cut away that, that top layer to look inside. And what you can appreciate is that all of it is alpha helix. And that's typically the conventional way that an alpha helix is presented in research. Before I move to the next structure, is there any questions about that? Yes. Sorry? Not the red. So forgive me, for the image to the right. So it's these like gray ribbons. So that's typically the notation for an alpha helix when looking at a protein structure. Yes. These are the protoporphyrin rings. And so for this exercise and trying to understand alpha helices, you can ignore the red structure. The important thing to note here is that hemoglobin is entirely alpha helices, and that this is the way alpha helices are represented when looking at protein structure. Yes.
Why are the alpha helices important? What are their functions?
So alpha helices. So these structures are. It's a way for us to understand how a protein is structured as a result of the hydrogen bonds that occur across amino acids. It's one of the structures. Another structure that we're going to discuss in a moment are beta sheets. But it's important to note that this emerges, that proteins develop this structure as a result of all side chains being stuck to the outside. And it also informs that if those side chains are hydrophobic or hydrophilic, how it's going to interact with its local environment, as well as the proteins around it. There are proteins that have a lot of alpha helices that are very hydrophobic, that are embedded within the plasma membrane of a cell. And we'll talk about plasma membranes in the next lecture. But how many alpha helices it has, and what amino acids are present within those alpha helices will help inform its function, because there are sites that are, for lack of a better phrase, activated, that can be activated, some amino acids, and others can't. And I think that was in the pre-lecture, amino acids that can be phosphorylated. We have serine, threonine, and tyrosine. And so proteins that have these alpha helices that are hydrophobic, that are embedded in the plasma membrane, that then have sites that maybe stick down into the plasma membrane that are hydrophilic, will potentially have amino acids that can become activated and elicit some downstream consequences, if that makes sense. So it's just a really helpful way of understanding the structure and the function of a protein. Any other questions? These are great questions, by the way. Okay. The next structure, the next secondary structure that's informed by hydrogen bonding is beta sheets. Beta sheets are typically a result of hydrogen bonding that occurs across a bit of a greater distance. What you can see is that there's, again, hydrogen bonding being denoted by the dotted line across the carbonyl group up to the top, down to the hydrogen off the amine here. And what's important to know is that when we're looking at the alpha helices and where the side groups were present, they were all sticking outside. But with the beta sheet here, what you can see is the side chains being denoted by the R. You can see that they go up and down, up and down, up and down. If you look across the peptide, across the peptide, and this creates like a bit of a corrugated structure that is what makes up the beta sheet. And so one thing that we'll do now is we discussed earlier how proteins or amino acid sequences are read from the N terminus to the C terminus. And so one exercise now that we're going to do is for the top amino acid group, or for the top row of amino acids, which side is the N terminus and which side is the C terminus. You can discuss with the people around you. I'm going to take this moment to get a drink of water, but I'll give us 1 minute, and I want you guys to try and see if you can figure out which side is the N terminus and which side is the C terminus. All right. Does anyone? All right. So N terminus. By a show of hands, N terminus on the left side. You can ignore the arrows. Okay. N terminus on the right side. Okay. Would anyone like to raise their hand and explain why? Yeah, go ahead. You said N terminus on the left side?
No, I said, sorry. I said I thought it was actually related to molecular type.
Mm-hmm.
And that is on the left side. So C is like a C double bond between
Okay. Okay. So what's important to know is that polypeptides, they're strung together from one amine group, one carboxyl group to the amine group. And so it's important to know that if we're looking for the amine group, so we're going to start with the nitrogen. So the nitrogen is always going to be first, is going to go towards the alpha carbon that has a side chain off of it. And so when looking over here, if we think it's on the right side, if we start the nitrogen, move over to that carbon does not have the side chain off of it. That's not the alpha carbon. So if we move over here to see if we're on this side, if we start from this nitrogen here and move down to that carbon, that's the alpha carbon that has the side chain off of it there. So this is actually the N terminus here. I understand that carboxyl group might be misleading, but again, this is the directionality of how amino acids work, which means that in a polypeptide, there's going to be a carboxyl group next to the bound, having a peptide bond to the adjacent amine group, but there's still a directionality. So what that means is that you need to look at the nitrogen here and look which way is the alpha carbon exists, which way is the alpha carbon. And so the alpha carbon is not to the left because it doesn't have the side chain off of it. If you look to the right, there's a carbon with a side chain off it. So that's the alpha carbon. Does that make sense? If it's any consolation, I got this wrong when I took this class too. So don't if you got that wrong, this is the time to go through these questions and figure things out. Yes. So that is an alpha carbon, but which way is it? Yeah, so that means there's the amine group. We're marching this way to that alpha carbon. That's a good question. So again, we're started. So a good way to look at it is, okay, I'm looking for the N terminus. I'll look for the nitrogen. Which way is the alpha carbon? Which way has the carbon with a side chain off of it? Because it's not that way, because that's the carboxyl group. So that means if I march this way, that carbon has a side chain off of it, which is implying the directionality. So if we look here, if we look on the bottom part, if we start with the nitrogen here, if we go that way, that has the carboxyl group. So that means if I march this way instead, that has the alpha carbon with a side chain off of it, which means this way is the directionality. This is the N terminus, and that's the C terminus. Does that make sense, guys? Great. So as I mentioned earlier, hemoglobin only has alpha helices. So to try and display what beta sheets look like in the convention that the way they're typically shown, we have here is proteins, a superoxide dismutase. And these have what we call anti-parallel beta sheets. And so typically what that means is this is typically how beta sheets exist in anti-parallel sheets. And so what you can see is there's a directionality again, where we can go up and down, but they exist in an anti-parallel fashion. Parallel beta sheets do exist, but are a bit more rare. So this is flavodoxin. I'm trying to remember what this is. This is an electron donor in a protein that's an electron donor in bacteria. And so it's surrounded by alpha helices, but embedded in the middle are parallel beta sheets. But again, those are typically more rare. And what you'll usually see are anti-parallel beta sheets. So the last set of interactions that we're going to look at for today, are there any other questions before I move on to the next interaction? Yes. Yeah. Any other questions? All right, so the last set of interactions to consider are hydrophobic interactions. So hydrophobic interactions typically are typically used as a way to try and understand how a protein will act in aqueous solutions if it's thermodynamically favorable to them to avoid aqueous solutions, hydrophobic, or to be in them if they're hydrophilic. So the goal here oftentimes is to try and reach thermodynamic equilibrium. So when thinking about hydrophobic interactions, that's kind of the backdrop or the context in which to consider these. And so if we took a bottle of water, and if we added pentane to it, that's an aliphatic molecule that has no electronegative parts to it. And if you shake it up within a bottle of water, what will first happen is it'll be caged off within different water molecules. But what this does is that this decreases the mobility of water, and it will decrease the randomness or mobility of water. And that randomness is called entropy. So in thermodynamics, you cannot decrease entropy. It only increases. We typically in thermodynamics, you say we cannot create energy. We can't. You don't create energy. Here, you want to increase entropy. That's kind of the backdrop of hydrophobic interactions. And so the hydrogen bonds between water is disrupted so that the pentane, this hydrophobic molecule, is then partitioned off and clustered together so that water, because pentane is not forming any hydrogen bonds with water, what's happening is that it's reducing the surface area that it has with water. So it's then cordoned off and clustered together. You can also think of it if a bottle of, if you have water and oil, olive oil, and if you mix it up, you first see all of the bubbles and it's partitioned within the water. But then over time, because it's thermodynamically favored, it then settles and is then partitioned off from the water. Does that make sense? So because there's no hydrogen bonding occurring, because there's no desire, because there's no hydrogen bonding, there's no desire for, it's thermodynamically unfavorable for oil or pentane to be in droplets through the water. It's thermodynamically favorable to decrease entropy and for it to have the least amount of surface area between it and water. So then it's partitioned off. Does that make sense? Hydrophobic interaction, yes. Yeah, so what that's trying to describe here is if it stayed in that top right, if it stayed in that top right format, if pentane was mixed throughout the water and cluster in different little tiny clusters or droplets, what that does is it is reducing the mobility of water and its randomness, or it's reducing its entropy, which means it cannot stay that way. Therefore, it has to go down to this new format. Does that make sense? That is confusing. I'm sorry about that. But yeah, so you want to decrease entropy, and so in doing so, because there's no hydrogen bonding occurring, it'll be partitioned off. And so typically, if you think of a hydrophilic protein, it'll have an electrostatic charge. There will be an availability for electronegativity, and it'll want to interact with water and have hydrogen bonding occurring between its dipole and the hydrogen on water. And then it'll be mixed throughout. If you think about salt, salt you can mix in water. Sugar does not want to mix in water because it doesn't have the same, it won't create the same hydrogen bonds if you think about the structure of sugar. Does that make sense? Okay. If there's any questions, please reach out. Okay. And so because of the high energies involved, the most important forces driving protein folding, this is one of the most important factors driving protein folding and formation in cells. And so the hydrophobic interactions are really critical to helping trying to understand what is favorable for a protein and how it wants to behave. And so if we look next at these aliphatic side chains, these will not have a charge. So they're going to be hydrophobic. And valine specifically is going to be important for us when trying to apply this back to sickle cell disease. And that's also true for most of the aromatic side chain amino acids, with the exception of tyrosine because it has that hydroxyl group sticking out on its side chain. But phenylalanine and tryptophan are also going to be hydrophobic. So when going back to hemoglobin, here we've highlighted the green amino acid residues, the hydrophilic amino acid residues in green, and the hydrophobic amino acids in purple. And if you think about hemoglobin and it being within a red blood cell, it's in the cytoplasm, which is an aqueous solution or aqueous setting. And so the hydrophilic amino acids are on the outside because it's favorable for them to want to be in and around water or in an aqueous solution because of the dipole interactions with the surrounding environment. Whereas the hydrophobic ones do not want to be around water. They're embedded inside the hemoglobin structure. So we've looked at the hydrostatic bonds. We looked at the electrostatic bonding that happens that kind of informs how the subunits come together. And so the hydrophobic interactions also inform what amino acids are on the outside or facing inward. So to put all of this together and apply it back to sickle cell disease. in sickle cell disease, what happens is we have a substitution for glutamate to valine in the beta chain of hemoglobin. So there's a nomenclature for how we typically have a shorthand for these mutations, whether if they happen in disease or if we make them happen in a lab. So in beta chain hemoglobin, at position 6, we'd say it's glutamate to valine at position 6, E6V. So it's going from this to that. Does that make sense? And so if you remember, glutamate is hydrophilic, and so that's why it's on the outside, as we looked in the previous slide of where the hydrophilic and hydrophobic amino acids are. But valine is hydrophobic. And so right now, because it's substituted for a hydrophilic, we have a hydrophobic amino acid substituting for a hydrophilic one. That is not thermodynamically favorable. So what this protein will do now is that it will change its structure to accommodate for that. It will try and bend over and embed that valine now so that it's not available. But what's important to know now is that that's not the only valine that's in the protein. So now, because it bent and tried to bear that valine, there are other valines that are highlighted here that are now sticking on the outside. So again, it's thermodynamically favorable to want to try and bury or cover that hydrophobic site. And so what begins to happen in sickle cell disease is that these hemoglobins begin to form fibers that then stack on top of each other within the erythrocyte. And so if you remember at the very beginning of this lecture, erythrocytes are pliable. They're able to squeeze through the thin capillaries into distal sites or even within the lung, the important site where oxygen exchange occurs. But here within the erythrocyte that has this mutation, these hemoglobin fibers will begin to layer and stack on top of each other. And what ends up happening is it alters the entire mechanics of the erythrocyte as well as its structure. And it buckles and has this sickle shape because of, if you look in the bottom right image, these hemoglobin fibers begin to just layer and stack on top of each other. And when the hemoglobin is not able to be pliable and fit through capillary spaces, it creates these occlusions in capillaries that then creates the chest pain that we talked about, that then also creates the dactylitis, the swelling and inflammation of the digits. And so the treatments that are available for sickle cell disease, the first set are palliative, are meant to just mitigate some of the pain and mitigate some of the discomfort. Fluids, pain medication, oxygen therapy. There's one interesting treatment, hydroxyurea, that upregulates fetal hemoglobin. Fetal hemoglobin, so if we remember from the beginning, hemoglobin is a heterotetramer. That means it has 4 subunits to it, and they're a combination of different kinds of subunits, 2 alphas, 2 betas. Fetal hemoglobin actually is 2 alphas and 2 gammas. And so what's important to know here is that fetal hemoglobin is what is present during development, and it's actually able to mitigate the sickling of erythrocytes that occurs. People will also receive, yes? Yeah, so during development, so that's a good question. During development, you're going to have different genes. So alpha is a different gene than beta, which is a different gene than delta and epsilon. And the way this works is that during development, there's primarily some genes that you'll have red, that you'll have some genes that are turned on or red, that then do not exist later in life. Or you'll have some genes that are not on during development that will then be turned on later in life. And so we'll talk about this later when we cover the immune system. Most kids, when they're born, do not have a fully developed immune system, do not have, I did my PhD in molecular immunology lab. And so like for me, like this is where I kind of go back to. There are genes that are on or off in development that are not on or off in adulthood. And so what's been discovered is that providing an upregulation of fetal hemoglobin helps to resolve the sickling and sickle cell anemia. And so there have been also bone marrow transplantations that have been trying to remedy this. But what's in an effort to try and update the class a little bit, there's a really interesting clinical trial that was, I think, just started this year, where we'll learn what CRISPR-Cas9 is later on. It's a gene editing tool. And it was discovered that there's a specific gene that regulates fetal hemoglobin. That's called BCL11A. And what they're able to do is it suppresses fetal hemoglobin. So what they do is they try and knock out that BCL11A gene, and then that allows the upregulation of fetal hemoglobin that resolves the sickling and sickle cell anemia. And so, when I took this class 13 years ago, we didn't know anything about BCL11A. We just started having what's called a GWAS, genome-wide association studies, where you just try and look at what mutations cause specific traits across the entire genome. And in the last 13 years, we've been able to go from advancing those studies to identifying this gene as regulating fetal hemoglobin to now being able to edit the gene in people and resolve sickle cell disease. This was published in the New England Journal of Medicine last month, I think. And so there's a lot of excitement because, again, this is a monogenic disease that's inherited and recessive. And so the idea is that potentially with first trying to use gene editing tools to address monogenic diseases, this could be a really powerful way to help resolve that specific, the mutation in beta chain of hemoglobin to resolve or mediate the sickling. And when we talk about the evolutionary advantage of different things or trajectories, I mentioned earlier that this is primarily in African Americans, or particularly those from a West African background. And the question is, why is that the case? This was not selected for in other populations. Why was it specifically selected for in this population? And it's and to help make sense of this, HB is hemoglobin. Remember, we have two chromosomes or two copies of everything. So typically you'll have it as a superscript above for the two copies. So A is the traditional allele, S is the sickled allele, and it's recessive, so you only need one to get the disease. So what's been identified is that having that mutation provides protection against malaria, and so it appears that primarily in West African populations where there is a large incidence rate of malaria, it appears to confer tolerance and protection for those that have this disease. And this study was, I think, just done 10 years ago. Yeah, 2011. And so we're still trying to figure out ways to apply this knowledge. And this is also meant to try and help demonstrate to you all that the things that we're talking about in this class, we're still finding ways to address it, and we're still better understanding it. And the projects that we're going to be doing in this class are meant to provide you guys opportunities to think of new ways and new opportunities to take new tools like CRISPR-Cas9 that have been developed and apply it in new creative ways. And finally, if structure, one of the things I want to try and do is, if any of the lectures really fascinate you, like for me, I had my moment as an undergrad thinking angiogenesis sounds amazing. How can I learn more about this? There's a fantastic group of researchers at UVA. This is just a quick snapshot of them, of who looks at structural biology, and I know the two on the ends are really active researchers at UVA. that provide opportunities for undergrads to get plugged into research. So if any of this interests you, I definitely recommend you reach out to these people for research opportunities. Are there any questions? Yes. That's a good question. So to upregulate something means to express it more. To downregulate means to express it less. So there are different ways of controlling upregulation, downregulation in biology. I mean, you can do it artificially, or there's kind of feedback loops that naturally occur, where if a protein is being released and secreted, there's kind of a break to downregulate it naturally that then inhibits it. And so there's different ways of accomplishing upregulation, downregulation, but it typically means the expression of some gene or expression of some protein. Yes. Mm-hmm. Yeah. So that's a good question. For each amino acid, there's a 3-letter naming system, and there's a 1-letter naming system. So for glutamate, it's E. G is glutamine. That's a good question, and it's easy to confuse those two. Any other questions? All right. With that, that concludes today's lecture. Appreciate your questions, and I'll be here if you have any further questions.